I want to say hello for foreign participants from Italy, Egypt, Costa Rica, Ecuador, and Ukraine, and also to participants from Romania. This is the second edition of AlgaTech. First edition was last year. We have some rules to be everything okay. First rules is to keep the microphone off and video on. In this way, you can understand everything it's going here, and we cannot intervene in the presentation. Each presentation will have 10-15 minutes and 5 minutes debate. If we succeed the presentation and are more discussion, we can make after. We have a debate section, and all the question get put on chat, and each one will reply to the question from the chat if we will go more than 5 minutes. Idea is to keep running the event and to can share the information. This event is made by Institute of Research and Development for Aquatic Ecology, Fishing and Aquaculture from Galati, Romania, in auspices of Academy of Agriculture and Forestry Science Romania, Gheorghe Ionescu, Șișești. The first will be Mr. Nicolae Patriche, teacher from the organizer community. Mr. Patrick is also correspondent member of Romanian Academy of Agriculture and Forestry Science, Gheorghe Ionescu, Șișești. Mr. Patrick, please.
First of all, I want to say welcome to everybody on behalf of General Director of the Research Institute of Galati, Professor Floricel Maricel Dima, and of course the whole staff. Benvenuto per nostri amici di Italia. Ahlan wa sahlan for our colleagues from Egypt, and welcome and also bine ați venit for all colleagues from Romania. We want also to thank you very much for the leadership of Romanian Academy of Agriculture and Forestry Sciences, to Vice President Dr. Aurel Florentin Batiu, and for the secretary of the section, Professor Nicol Petculescu. Also, we want also to thank you very much to our colleagues from research station from Nucet, and also for the colleagues from the University of Galati. Welcome to the fascinated world of algae. My word in this is very short, and I wish all success to everybody. Thank you also to the president of Romanian Academy for Agriculture and Forestry Sciences, Professor Ioan Jelev, who joined us. Thank you again. And I wish to all participants a good session for today. Thank you very much. Mihai, you have the floor.
Thank you very much, Mr. Patrick. The second speech will be given by Mr. Ioan Jelev, President of Academy of Agriculture and Forestry Science Romania, and also President of European Academy of Science applied to Agriculture, Food and Nature for this period from Science 2024 to 2026. Welcome, Mr. Jelev. You have the words.
you very much. It's a pleasure for me to take part at this very important international meeting. Also, I wish to send many greetings to our colleagues from Egypt because we were very much involved in the cooperation in this field with Egypt, a successful cooperation. And further, I apologize, but I will speak in Romanian due to the fact that a lot of participants are from Romania. Deci este, domnule profesor, o mare plăcere pentru mine să aduc pe domnul profesor Patriche, de care mă leagă și o veche prietenie și am avut plăcerea ani de zile să lucrăm încă de pe vremea când lucram în Ministerul Mediului. E o mare plăcere și onoare pentru mine să iau parte la un astfel de o dezbatere internațională importantă și profit de acest moment ca alături de colegul meu, domnul vicepreședinte Aurel Badiu, să vă transmitem și din partea conducerii a prezidiului Academiei felicitări și domnului director general Maricel Floricel Dima pentru această importantă dezbatere. Institutul pentru noi este unul din institutele, să spunem, de referință în domeniul cercetării. Vă doresc foarte, foarte mult succes și am încredere că se va derula bine și această evaluare care vă așteaptă în acest moment a performanțelor în domeniul cercetării. Este un moment de referință și așa ca o încurajare vreau să vă spun că până în prezent unitățile noastre de cercetare care au trecut deja prin acest pas s-au prezentat cu cinste, mai ales în contextul situațiilor cu care ne confruntăm din punct de vedere al finanțării, care lasă încă mult de dorit pentru a putea asigura o cercetare performantă. De asemenea, eu aș dori să subliniez importanța tematicii și dezbaterilor care vor urma în cursul zilei de astăzi. M-am uitat și pe programul acestui seminar și simpozion internațional cu teme extrem de importante și am dorit în mod deosebit să am această șansă de a participa la acest moment științific de referință, întrucât domeniul respectiv mă amintește așa cumva de preocupările pe care le-am avut de-a lungul anilor și în cadrul Ministerului Mediului, dar și ca implicare în domeniul apelor. O perioadă destul de îndelungată am fost șeful departamentului apelor și încă de pe vremea aceea am colaborat foarte mult cu institutul. Domnul profesor Neculai Patriche era deseori oaspete și invitat special la Ministerul Apelor, Pădurilor și Mediului și totdeauna m-a impresionat, să spunem, această lume a apelor în care vă desfășurați activitatea și cred că este chiar un domeniu extrem de important de viitor, iar prin implicarea în colaborările internaționale, iată că institutul se ridică la un nivel deosebit de înalt, de colaborare științifică, activitate intensă, accesarea unui număr foarte mare de proiecte și programe și cred eu că în acest fel și acest moment al evaluării va fi parcurs cu bine de acest institut de referință. De asemenea, îi salut și pe ceilalți colegi care lucrează în domeniul pisciculturii și pe colegii de la stațiunea de la NUCET și, în final, încă o dată vă doresc foarte succes, dezbateri fructuoase care să aducă, să spunem, anumite clarificări și dezvoltări viitoare în acest important domeniu. Încă o dată felicitări și succes organizatorilor.
Thank you very much, Mr. President. I will make a short resume for the people who don't understand what the president say in Romanian. For short, Mr. Zhelev say it's a great pleasure to be here at this debate. He want to send congratulations to all and good luck. The institute who is involved in this moment go to a referential moment, and he hope this debate and the thematic of the debate will help us in the future. Also, it's very important moment for science because Mr. Zhelev in his past was involved in the water sector, and he make a lot of collaboration with World of Waters, and for his opinion, this is important area. Also, it's important to go to program like that and project like that. Also, he want to salute all the participants and wish good luck for debate. This is the small resume for the people who don't know Mr. Zhelev's speech in Romanian. We go with Mr. Aurel Florentin Badiu, who is the Vice President of Romanian Academy of Agriculture and Forestry Science, Gheorghe Ionescu Șișești, and also Mr. Badiu is involved in a lot of projects. Mr. Badiu, you have the words.
Good morning. Unfortunately, my English is not very good. I continue in Romanian. Sunt deosebit de onorat că m-ați invitat la AlgaTech. Sigur că da, specializarea mea de agronom în zona cercetărilor privind algele este destul de limitată, însă curiozitatea este mare și aș încerca să fac cu domniile voastre în speech-ul meu, în intervenția mea, o scurtă trecere în revistă a interesului agronomic pentru algele de apă dulce. De ce algele de apă dulce? Pentru că cu voia lui Dumnezeu avem ape dulci suficiente în România. În ultima vreme a apărut și o lege a pisciculturii în care nu cred că a intrat și domeniul algaculturii, dar asta nu înseamnă că nu trebuie să ne îndreptăm atenția în mod deosebit către alge, a căror utilizări ecologice, economice și economice sunt aproape de a fi de top, mai ales în contextul schimbărilor climatice și a modificărilor apărute în habitatele agricole. Sigur că da, toată lumea probabil se gândește la ce pot face algele de apă dulce. Cred că primul, în afară de a fi folosite ca indicatori biologici ai calității apei și ca, principalele organisme vii care oferă apei oxigen, de care au nevoie aproape toate speciile acvatice, prin fotosinteză fiind notoriu faptul că odată se produce oxigen, dar se fixează și dioxid de carbon. Esențial atât pentru fauna acvatică, dar mai ales esențial din punct de vedere al fixării dioxidului de carbon ca unul din gazele cu efect de seră major. În ceea ce privește agronomii, cred că bioremedierea ca modalitate de utilizare a algelor de apă dulce este esențială, algele de apă dulce fiind cele care pot absorbi nitrații, care se scurg spre pante în chiuvetele lacurilor și iazurilor, dar și în apele curgătoare. Sigur că da, nu este de neglijat absorpția de fosfați și de metale grele. Cred că va trebui să ne îndreptăm atenția în mod deosebit spre aceste alge de apă dulce. Sigur că da, majoritar în lume se utilizează algele și se studiază algele de apă sărată, însă caracteristicile principale acestor alge este odată posibilitatea de a le folosi pentru bioremediere și aici area de aplicație este mare, de la soluri poluate la ape uzate până la soluri contaminate cu metale grele, hidrocarburi și nu este de neglijat pesticide, cu atât mai mult cu cât fitobioremedierea transformă poluanții și îi elimină. Credem că o parte dintre, sau cred eu, că o parte dintre domniile voastre, mai ales cei tineri din zona Institutului de la Galați, s-ar putea îndrepta într-un viitor mai mult sau mai puțin apropiat în această zonă de studiu a microalgelor verzi și, într-o oarecare măsură, a microalgelor roșii, eventual pentru o creștere controlată a lor și utilizarea lor în afara, haideți să zicem, lucrărilor experimentale sau experimentărilor și intrarea lor în zonele agricole, în zona fermelor agricole, dat fiind faptul că pe lângă fixarea de azot directă în sol a celor care sunt autotrofe, dar pot să aducă o serie întreagă de beneficii asupra texturii solului, a structurii solului, dat fiind faptul că textura și structura solului suferă extrem de mult în perioadele de arșiță. Este notoriu cunoscut că în unele țări din subtropicale algele au început să fie utilizate în activitatea practică, taman sau exact în zonele în care arșița spune cuvântul. Nu mi-aș dori să ajungem în această situație, însă așa cum pare că evoluează agroecosistemele, viitorul este a unor agroecosisteme, dacă nu deșertice, aride în mod cert. Sigur că da, din toate punctele de vedere, putem discuta despre multe aspecte privind macroalgele de apă dulce, care pot să, am zis, fixeze biologic azotul, dar să și stabilizeze structura solului, să prevină eroziunea, pot degrada poluanți organici și, în mod evident, îmbunătăți fertilitatea globală a solului. Dar cred că am început să vă plictisesc, așa că mă opresc urându-vă succes unui program extraordinar de interesant, care, chiar dacă nu se adresează unui agronom, trezesc interes. De asemenea, țin să le mulțumesc invitațiilor dumneavoastră și din țară, dar mai ales celor din străinătate, pentru că se întrevede o dezvoltare serioasă, consolidată și sunt convins că în mod cert solidă a cercetărilor privind algologia și de apă sărată, dar sper eu și de apă dulce. Mulțumesc frumos și dacă v-am plictisit, să rămână. Asta este, sunt poluant, mă tratați cu alge. Succes lucrărilor conferinței.
And short, for the people who don't speak Romanian, Mr. Badiu, who is vice president of our academy, told something like that. It's honored to be here because it's an agronome, and he worked in agronomy research, and he thought his curiosity is very high to be, and for this reason is present here. He want to make some point about fresh dulcical water algae. In this context of climate change, he saw very important the algae, first because they are the quality water indicative, and second because through the photosynthesis will be produced oxygen and fixate the CO2. Also, they can work algae like bioremediation. It's very essential in this area because they absorb nitrate and phosphate and also heavy metals. Mr. Badiu sees some possibility in bioremediation, soil and waters using algae. Also, he hoped some of my colleagues from the institute from Galati to look to this area for research, and also to go to red and green microalgae, and to implement some project in agriculture. He hoped to don't be in the future so many area with dry land, but probably will be like that, and the algae can help fertility in this area. Also want to thank you for all participants and to help for future research in this area. This is the small short resume of what he said in Romanian. The next on the line will be Professor Nicole Petculescu, Secretary of Food Industry Section from Academy of Agriculture and Forestry Science, Gheorghe Ionescu, Șișești, Romania.
Hello to all the participants from the part of the Food Industry Section of the Academy of Agricultural and Forestry Sciences. It's an honor to meet you all the participants to this scientific symposium dedicated to algae. Hello Egypt, hello Ecuador, hello Costa Rica, hello Italy, hello Ukraine, and hello Romanians. Allow me to bring the salute and welcome to this symposium from the president of our food industry section, Mr. Professor Petru Niculiță, and also from all our members of the food industry. One of them, of course, is here online, Mr. Professor Neculai Patriche. So this is the second edition of the scientific symposium concerning the algae, very well entitled Algae as a Sustainable Resource for Food, Health and Industry. So practically we respond why algae? Because they are found in aquatic environments, including saltwater, freshwater, brackish water, but also can be found in dam surfaces, soil or snow. Are vital to aquatic ecosystems, and also they are a major producer of Earth's oxygen through photosynthesis, so are crucial also for the global carbon cycle. Are used in environmental monitoring to assess water toxicity, and also we have to focus on the economic utilization of both marine microalgae and also macroalgae, because they are used for a long time as human food, animal fodder, a source of bioactive products like oils, polysaccharides, for biofiltration, for soil conditioners, and in waste treatment and aquaculture. In fact, all the papers which will be delivered today will present a part of algae assets. The cultivation of algae is presently one of the most productive and environmentally friendly forms of livelihood, among the coastal population. In conclusion, the beautiful world of the algae is a very interesting domain for scientific researchers, for food industry, and of course for our planet. Allow me to wish you success to all the participants to this scientific symposium dedicated to algae.
In business, first
Yes.
presentation, it's cultivation of Arthrospira platensis under Black Sea conditions from National Institute of Marine Research and Development, Grigore Antipa from Constanța.
Okay, thank you. Second, please. My name is Pantelena Daniela. Today I will present some results from our project, Exacaqua, namely cultivation of spirulina or Arthrospira platensis under Black Sea conditions. I will start presenting some objective and overview of our project, spirulina platensis uses and applications, media cultivation conditions in our lab, cultivation systems, monitoring growth performance, harvesting challenges and conclusions. As you know, probably know Exacaqua aim to improve numeric capacity in algae aquacultures, focus on nutritional rich and ecological value species, test different cultures system for spirulina platensis, but not so. We also growing chlorella, but we are not presenting today these results. Supporting development of scalable, sustainable cultivation technologies and adapt and optimizing biomass production in Black Sea conditions. Here are presented some uses and application for for spirulina. As you probably know, spirulina is a microscopic filamentous cyanobacteria. Its growth is strongly influenced by light intensity, carbon dioxide, bicarbonate availability, and nutrient concentrations. That conditions, they actually influence their morphological aspects. The strains tend to elongate or spiral according to their conditions. As key uses and applications, they are used mainly as nutritional supplements in medical and therapeutic uses. They are used in agriculture and animal feed, in cosmetics and skincare, and also in environmental and industrial applications such as biofuel or wastewater treatment, and also carbon assimilation industry. Median cultivation conditions. In our lab, we use it. Initially, we started to grow in rich seawater medium and in spirulina medium. That medium were bought from the firm that we from where we buy the strains. I'm sorry, I have a little emotions. Okay. After that, the spirulina will grow in the sterilized and filtered seawater and also in tap water. After that, we added, we grow it into two different media. One media was with the F2 media, and the other one with the Zaruk. The laboratory conditions were approximately 20 degrees. We furnished the air bubbling. As a cultivation system, we started with the test tubes. After that, we grew it in the 50-100 milliliters, and after that, we started progressively growing the microalgae, the spirulina, until 5, 10, or 25 liters. In the left of the corner, you will see the how it looks, the spirulina, the platensis that we are growing is a straight filament. We don't have the spiraled one. The same conditions are growing, the temperature 20 degrees with a 16 to 8 hours of light as a dark cycles. As a monitor, we used visual checks, environmental control, and growth tracking. If the color of the culture is dark green, actually it was we consider that it's active growth. If it has a yellowish color, we consider to be a nutrient stress. And if it was a light green, we consider that it was a poor mixing or contamination of the culture. As I mentioned before, the condition for growing are approximately 20 degrees. And for the big culture growing in a large volume, we used air bubbling. At a pH of 9 to 10, and salinity according to the media. As a growth, we track the optical density and the dry weight. As a growth performance, you can see in the slides, we put some graphics when we present our results. In the 5 liters photobioreactor, you can see that the best performance was seen in the sterilized seawater plus F2 media. In the 10-liter alga bag, the best performance was also in the sterilized seawater plus Zaruk media. And in the 20 liters, 25 liters alga bag, it was the growth performance was best in the sterilized seawater plus Zaruk. Also, as a dry weight, the best results were seen in the fresh water media and in the seawater, sterilized seawater plus Zaruk media. Overall, spirulina, freshwater spirulina, has the best growth in sterilized seawater plus Zaruk media. As a harvesting, first we decanted the culture. The culture was collected. After that, we filtered on the mesh. We obtained the concentrated biomass, the wet biomass, and after that, we dried by lyophilization process. As challenges that we have growing the spirulina, we have contamination issues, flagellates and foam during the aeration, biofilm buildup. We actually try to grow it at a higher temperature in an incubator, but it caused precipitation, discoloration, and growing instability. As infrastructure limits, we have growth chamber supposed only 1-2 liter vessel, and we have no aeration, but that's why preventing effective scale up at the higher temperature. And harvesting bottleneck, we have time-consuming multi-step infiltration and biomass recovery. As a conclusion, significant differences were observed across media, with sterilized seawater and Zaruk media supporting the highest and the most sustainable biomass accumulation, followed by freshwater media. These species can be successfully cultured in closed photobioreactor systems using seawater-based media, provided that alkalinity and carbonate availability are optimized. This highlights the importance of medium composition and salinity control for maximizing biomass productivity in regional conditions. The result confirmed the possibility of developing scalable spirulina platensis cultivation in Romania and provide a practical basis for future pilot and industrial scale application in the Black Sea Basin. Thank you. That was my presentation.
Next, Mr. Vincenzo Longo is here, and we'll keep the presentation nutritional and functional foods. Mr. Longo and all the collective are from Institute of Agriculture, Biology and Biotechnology, CNR Pisa, Italy, and HER Research Group, Department of Biotechnology, some of Cristina Chivera and Carla Sandoval Guano. So, Mr. Longo, please put the presentation.
Thanks, the organizer of the meeting, for inviting me. It's a very interesting day because algae. Can you hear me?
Yeah,
Yes.
perfectly.
Okay.
We hear you.
Algae are very important products that can be used also for food, for cosmetic and other things. It's very nice to join with you this day. I try to share my presentation. I would like to talk before about nutraceutical and functional food because algae can be considered functional food. And I am Vincenzo Longo from Institute of Agriculture, Biology and Biotechnology of the National Research Center of Pisa. I would like to thanks also Professor Nicole Petculescu because I know her very well. I was in her university and we met in Pisa just some couple of weeks ago.
Thank you very much.
Nutraceutical are the bioactive compounds that can be present in food and in plants, and usually are very important because they can be important to help us in the prevention and the treatment of disease. And nutraceutical is different because there is confusion sometimes between nutraceutical and functional food, but nutraceutical is different from functional foods. And it coined from nutrition and pharmaceuticals, be in the form of capsules, powders, or fortified foods. Example of nutraceutical can be omega-3 supplements, resveratrol, lycopene, curcumin, and so on. A functional food is a conventional food that has been enhanced with additional ingredients or naturally contains components that offer health benefits beyond basic nutrition. Functional food can be consumed as part of a normal diet. They are very rich in bioactive compounds such as vitamins, minerals, fibers, probiotics, and so on. Yogurt with probiotics can be considered a functional food, or fermented food can be also considered. And also algae, my idea is that can be considered functional food. I would like to say just something about fermented food because we have my research group since 20 years ago start to work with fermented food and to see the health effect of this. Because functional food are fermented food are very important and are very rich. They have both prebiotic and probiotic and postbiotic potential because are rich in fibers and in microorganisms. And it was demonstrated also that high fermented food diet can go to improve our macrobiota. You know that in our intestine are present microorganisms, a consortium of microorganisms, and that is called macrobiota. But macrobiota is very important because there is a connection with brain, with liver, with immune system, and it's very important the food that can you to increase the biodiversity in our intestine. And fermented food was demonstrated in this review. You can see here that it was right just some 2-3 years ago, and high fermented food can go to increase the biodiversity, and go to decrease inflammation, cytokines, and go to decrease also oxidative stress. And we know that oxidative stress and inflammation can reduce the biodiversity in our intestine. We demonstrate just with a power weight of grain that some components of this that is called lysozyme G can reach our cells, and we know that we have an equilibrium between in our body. There is an equilibrium between oxidant and antioxidant. When we have a lot of oxidant, we have free radicals, and there is not equilibrium. Our antioxidant system cannot eliminate toxin. In this case, we have oxidative stress. But we know also that a nuclear factor called NRF2 that is present in cytoplasm of cells usually go to the nucleus and go to activate our antioxidant system. And we demonstrate some components of lysozyme G, maybe bioactive compound, antioxidant, go to activate this system, create a dissociation between NRF2 that is bound to KEAP1, NRF2 go to the nucleus and go to activate hemosigenase, the deaphorase, phase 2 enzyme, and antioxidant enzyme. This is very important. And we demonstrate also that lysozyme G can protect liver from toxicity of pollutants, but also we demonstrate that it's able to go to improve our microbiota. And this is important results. We published a lot of paper about this. But today we talk about AlgaTech meeting, and I would like to show also some results about this. Algae and microalgae are very rich in bioactive molecules that are very important for human health. Algae has emerged as a promising food source due to their high-value ingredients because they are very rich in proteins, as well as for their environmental sustainability and abundance. And we published some paper about this. I show just some of this because we, in collaboration with professor from Ecuador, Professor Cristina Echeverria and Carla Sandoval, that she will talk after, we have studied some microalgae from fresh water, and we demonstrate that are very rich in phenols and other antioxidants. But also after I show one paper in particular, because you have no time to describe all of this. But we demonstrate also with chlorella, another important, we published just now this, that was able, chlorella, to prevent to improve the COPT in using mouse model in pulmonary. All chlorella reduced in this paper with chlorella vulgaris, and we demonstrated the potential preventive effect of chlorella vulgaris using a model mouse with COPT, obstructive pulmonary disease. Okay, in particular, I would like in 3-4 minutes to talk about a particular microalgae, Chlamydomonas augaliformis, that we published about this, and we demonstrate in collaboration also with Echeverria and Carla Sandoval from Ecuador. We demonstrate that from Ecuador they characterized and morphologically and molecularly particularly, they identified this algae, microalgae, as Chlamydomonas aegliformis. And we studied for nutritional and nutraceutical properties. We went to measure the phenolic composition, the antioxidant capacity, and the fatty acid profile of these lyophilized cells were determined. We also used a cell line, human microvascular endothelial cells, to study the capacity of this Chlamydomonas aegliformis to reduce the toxicity induced by LDL, oxidized LDL that go to mediate oxidative stress. And we demonstrate that this microalgae is very rich in flavonoids, phenols, flavonols, and also it demonstrates a very important antioxidant activity measured by different methods, HORAC, FRAP, DPPH. When we go to use cells, we have control cells here in this graphic. We have control cells, the cells after treatment with healthy oxidized LDL, that they go to create oxidative stress and increase inflammatory markers like interleukin 6. But and we studied this interleukin 6 at gene level and protein level here. And after pretreatment with Chlamydomonas aegliformis, we found a protection here, but more at protein level. You can see that there is a very reduction of oxidative stress. And this is very important. We demonstrate this, but also we went to see some other enzyme like hemocygenase, DT-deaphorase, and there was also a protection. We can say that based on the outcomes of our in vitro study, Chlamydomonas aegliformis demonstrate to be a promising source of bioactive compounds possessing antioxidant capacity, and we could consider this microalgae as prospective functional food. But there is another review where some authors demonstrate that the utility of algae as sources of high-value nutritional ingredients, particularly for alternative complementary proteins to improve human health. And you can see in this other slide the papers, all of different algae, green algae, brown, red, microalgae, and macroalgae, and some of these are considered by European Union as novel food. I finished my presentation and thank you for your attention.
I just want to say congrats to Mr. Professor Longo and to his performance team for such a beautiful paper. We discovered that algae can be used not only as a functional food but also as nutraceutical because they have a medical effect, like Chlorella vulgaris was demonstrated in his paper. They have antioxidants, antibacterial, and anti-mutagenic activities. And also, thank you very much for your study case regarding Chlamydomonas aegliformis. And also here we understood its fantastic values like nutritional and nutraceutical properties. And also the fact that generally speaking, the algae are a very wide and a very wonderful universe, and they are very useful for human food and human medicines. Thank you very much, Mr. Professor Longo, and your fantastic team.
Thank you. Thank you, Nicole. Thank you.
Thank you very much, Mr. Longo. We will try now with Mr. Darwish from Egypt, if it's ready.
Okay, good morning, everybody. I'm very happy to be with you today. my presentation about maximizing the production of the natural pigments in Spirulina platensis by changing media composition. This work is the result of scientific collaboration with the Romanian side from Institute for Research and Development in Aquatic Ecology, Fishing and Aquaculture in Galati, under supervising of Dr. Dima and Alina, and from Egypt, Dr. Fahad Abdullah and Dr. Hassoub and Dr. Hoda. Yes. The project objective, modifying the media composition to achieve maximum pigment production from Spirulina platensis and utilizing alternative solvent to enhance the efficiency of pigment extraction from Spirulina platensis. Yes. Pigment is an important food additive that play a major role in the sensory impact of food, and algae is an important source of natural pigments and contain chlorophyll, phycoerythrin, and carotenoid and other natural pigments which be very important. Besides staining, the pigment also have powerful physiological activity such as antioxidant, anti-inflammatory, and anti-obesity and lipid lowering. We start the cultivation of spirulina in the lab. Please, another. Next, yes, next. We start spirulina platensis isolated from Egypt, from soil and from water, and after that it's cultivated with heterotrophic condition based in the rock media. Under three conditions. First one, salinity stress and nitrogen starvation and high intensity of light. Yes. Algal scaling up. Several experiments were conducted to increase the amount of algal mass production, and stress experiments were conducted to increase the amount of pigment under nitrogen starvation and salinity stress and high intensity of light. This is the unit which we use under plastic container. And this is the final product of green spirulina, and under stress it become yellow color. We will see it now. This is one. Dry weight. The initial of dry weight is become increase about 250%, and also the carotenoid. I'm sorry, there's some problem with the presentation. And also total coffee was increased about 250%. Total chlorophyll is increased about 272%, and total carotenoid increased about 221%. And after that, we start to extract the pigments extraction. The best extraction was due to using methanol and dimethyl sulfoxide. It's about 160 milligram per gram as dry weight. and in dry weight it has become more better than fresh weight. For carbonated extraction, the best result with methanol also with dimethyl sulfoxide and methanol, and also it has become more better with dry extraction. The determination of phycocyanin with spectrophotometer, it's about 4.8 milligram per gram from Spirulina platensis. Also, phycocyanin extraction from spirulina under dry weight and fish weight, under dry weight, it's become more better.
This is all. Some conclusion, Mr. Dawish.
Yes, okay. The conclusion, phycocyanin could be produced with high amount from Spirulina platensis under specific condition. Salinity stress and nitrogen starvation lead to increased carotenoid accumulation and lipid content. Also, pigment extraction method affected extracted pigment quality, and dimethyl and methanol were best solvent for extraction maximum pigment from Spirulina. But if I will use it for food additives, I will use acetone extraction to be more safe. And we're fortunate to have a scientific collaboration with an outstanding working group in Romania. In this coming period, there will be greater cooperation with the great Romanian side to maximize the research outputs and outcomes. Thank you very much, and thank you for you, Mahai.
Okay, sorry for these technical problems, but we understand what you say.
I am very sorry for this problem.
Yeah, no problem.
Welcome.
If are some questions.
Nice to meet you. Okay.
Some questions are? No. If are not, we go to the next presentation. Highland microalgae and cyanobacteria, exploring the biotechnology potential of Andean freshwater ecosystems. Carla Sandovan Guano, Santiago Zarate, and Vincenzo Longo.
It's fine. Okay, thank you. Good morning to everyone. I am Carla Sandoval. It's a pleasure to share with you our work in the Universidad Técnica del Norte. I will present, the title of my presentation is Highland Microalgae and Cyanobacteria, Exploring the Biotechnology Potential of Andean Freshwater Ecosystem. So I would like to start my presentation with a little introduction about the place that this research was done. This is the province of Imbabura. It's called the province of the lakes. This is located in the north of Ecuador. So this province is so important because there are many ecosystems and important landscape with geological importance. So for that reason, some years ago, UNESCO named to Imbabura as a worldwide geopark. So in this province, there are many lagoons and lakes, but in this research, we are going to talk about two specific lakes, Gheorghe Lake and San Pablo Lake. The first one, Gheorghe Lake, is a Quechua name. It means jaguar, blood, and Cucha Lake, blood of lake. So this lake is his elevation. This elevation is above 2,000 above the sea level. And the second lake is San Pablo Lake. This is a volcano origin located in Imbabura volcano. The elevation is above 2,500 meters above the sea level. So this is the spot lake that we are doing the research right now. And this is the this is our start. In this context, what is the problem? Why we are doing this research? First, the lakes are essential resource for the communities surround them. Some agricultural and tourism activities are carried out without environmental regulations. This situation is becoming worse because the lack of the public policy to protect these ecosystems. So also, this is very little information. There is very little information to support the conservation of these lakes. So 8 years ago, our team started this research about what is inside of the lagoons, of the lakes, what is the importance of this ecosystem for our province and also for Ecuador. So we started our investigation, but this is our process. How we do that? First, we try to do the isolation and go through the lagoons on the lakes. And we right now we standardize some methodologies to take the sample and then go through the laboratory, and with using microbiological techniques, we try to isolate as much as we can, as much species as we can. So we have a little hermoplasm bank in our laboratory in the university, and we check these species under microscope to see morphological identification. And after that, we also performed molecular identification. And then when these species, we start the upstream process. We started to know, we want to know how is the performance of this microalgae under photobioreactors. So in these bioreactors, we try to scale up on 1 liter first, and then 3 and 5 liters. And we, in our team, we try to know the growth parameters as kinetic parameters, and also productivity and yields. Then we start with the downstream process regarding to the harvest process for us. In our lab, we did some biochemicals profiles regarding to protein, carbohydrates, and lipid profile. So the next slides, I will present some research conducting using high microalgae, which has been carried out through a multidisciplinary collaboration between our university and the other institutes, research institute in Italy, especially Institute of Agricultural Biology and Biotechnology National Research Council in Italy. So this is the first study what I will share with you. This study, we exploring the biotechnological application of this highland microalgae in these both lakes. We know as a result, we have here Pectinodemus pectinatus. This is a highland microalgae with the highest lipid content, high protein content, sorry. But we also find other 7 microalgae with really highest potential with lipid content, and also Ethlias alveolaris with the highest growth rate. So this is important for us because when we scale up, we want to know a highest growth rate microalgae. The next research is about this microalgae, Chlamydomonas agloiformis. This is we want to know about this microalgae, some bioactivity and some antioxidant properties and biochemical compositions. So this microalgae has a good nutrient and bioactive compounds that it is a potent antioxidant properties and protective effects on the human cells that suggest application or could be applied this microalgae for functional food and also nutraceuticals ingredients. This is the other research with Ethlia cerdiobularis, other highland microalgae. In this research, we study this microalgae revealing the strong antioxidant, antibacterial, and also antimutagenic activities with because attribute to the high polyphenolic content. So this microalgae has the ability to reduce oxidative stress in human cells. For that reason, we believe that microalgae could be applied as a functional food. This is other research focus on Pectinodesmus pectinatus and Ethlia cerdiobularis. This study, we want to know how microalgae respond to UVB exposure, focusing especially in the protective physiological mechanism. So in this study, Pectinodesmus pectinatus showed a strong resistance to the UVB exposure, and Ethlia cerdiobularis exhibit some specific acclimatization strategies related with the pigment recovery and also antioxidant accumulation to mitigate this photo damage provoked by the UVB exposure. The next one is also doing with Ethlia cerdiobularis microalgae, but in this case, this study is conducted by prove this microalgae as a biostimulant. In this case, this study researched the effects of these microalgae extracts and salt stress in 2 quinoa species. In this case, the application of microalgae extracts enhanced a bioactive compost that improving salinity resistance and increase the nutraceutical value on these both quinoa species. So this is a little summary of this research. As I told you at the beginning of the presentation, these research were done with collaboration, international collaboration, especially in Italy, and with our team biotechnology in Universidad Técnica del Norte. So we have a little hermoplast bank in our university. And also we have other species, but highland microalgae species that we have to improve the production, improve the yields, and also we want to start to doing some biochemicals profiles and also some others activities, biological activities, to know how it's the best a biotechnological application for this microalgae. So this is our future perspective. For our group is important link the research with cultural heritage in the Andean communities. As I told you at the beginning, these lakes is surrounded by indigenous communities. So it's important to know why this ecosystem is important for the people. And also we want to develop a baseline to raise awareness through the decision makers related to the water use, water management, and also conservation of the lakes. Also, we want to promote the research to provide other alternatives to sustainable use of this biodiversity. It's important to remind that there are a few research, few literature about this ecosystem in Ecuador. Also, there's a little papers related with the which is the potential of this highland microalgae, because most of the research in Ecuador and also around the world is related with marine microalgae. But this highland microalgae have a potential because these microalgae have to adapt to different altitudes, and also some of this microalgae is coming from of volcano origin. So this is interesting to understand all the physiological adaptation, and also it's possible to we want to know if this microalgae could be scale up and then produce good biomass with an industrial purpose. I would like to thank all my biotechnology group in Universidad Técnica del Norte, and also CNR in Italy, Pisa University, and International Society for Applied Ecology that support all these institutes, and many people that I want to thank, because as you saw in the presentation, there are many people doing research with our microalgae. So I would like to thank everyone. Thank you.
Thank you also. I have a question. Thank you, Carla. I have a question. I understand this lake are glacier lakes, no? They don't have river to put fresh water in?
Sorry?
These lakes, these two lakes are glacier lakes, no?
So, yeah, could be. We don't have real evidence of that, but yes.
But they are, the water, it's supplied by some local river or not?
Yeah. Local rivers, yeah.
So basically,
supply of.
It's a change of water in the lake. It's not the same water.
Yeah, change, but not so fast. It's really
so the consumption is bigger than the fresh water.
Yeah, and also the water of this lake maybe sometimes is used for agricultural purpose. So the water is not going.
At 2,600 meters, the weather is never it's not okay.
Yeah.
And the idea with this kind of microalgae, it's to make the research, and in the future to get a new probably economy in the region, to change a little bit the structure of monoeconomy probably.
Yeah, microalgae is not really a strong industry in Ecuador, because it's relatively new field in Ecuador. So as you see, the people, as you see, when the people see these lagoons, see only water. And the water is used for agricultural purpose, or maybe for mining. That is the other problem that we have right now, mining around the communities and around the lagoons. So we want to share with the communities and with the people that the lakes are life, and we can find a beautiful ecosystem in photosynthetic microorganisms, and also bacteria, and also other fungi as well, that you can use sustainable as a sustainable purpose, and you can give all these communities around surrounding the lagoons and economical
New
activities.
Yeah.
Perspective, yeah. But it's not right now. It's not only. It's not related like for this purpose.
Because I saw also the temperature, average temperature of the lake. It's a little bit low, 18 degrees. That means in the winter will be froze everything.
No. We don't have this.
So it's still water, not
water
ice.
Here. No, it's not ice. It really is a little warm here.
Okay.
You can see this temperature is more related at night, but in the day, in the morning, we have temperatures to 20 degrees sometimes. So we don't have ice in these lagoons.
Okay. Yeah, for me it's very interesting.
Nice.
Because everybody here discuss about algae at I don't know 20 degree plus or more, 30 degree, 40 degree, and you come with algae from the high altitude. So it's a little bit different about all the presentation from here. Okay.
Yeah, but these microalgae are right now are adapting to the laboratory to 20 degrees because this is
And the project, I saw a boat and some colleague of you from this kind of project. You have I don't know, subsidies from the government, or you apply to project, or no?
No, when we begin at the beginning of this research, we don't have really money to do that. We support with our money,
Biodiversity, yeah.
But to support. But we want to apply to local governmental fees to do more investigation related with the application of this microalgae, and also share with the people the importance to take care these lagoons and these ecosystems. So maybe the support, economical support coming from these different stakeholders around the surrounding of the lagoons.
Okay. If somebody have the questions, I finish my question.
Yes, I want to congratulate Carla and her team, and thank you very much because you allow us to discover the Ecuador, such a beautiful country, to discover your lagoons, your lakes, and especially your work. And what's very important is this multidisciplinarity and connections and internationality. And I think this is the key motor for the research. All the researchers from all over the world together and to try to do commune research projects for the benefit of the humans. Thanks a lot, Carla, for your work, and please, Michele, you have your floor.
Thank you. See you.
Okay. Thank you very much. We go to the next presentation.
Not yet.
Not yet. Okay.
Not yet. I want also to congratulate Carla for a very interesting paper about algae, which are developed up to 2,000 meters in Ecuador. In a special place, and in my opinion, it's very interesting what is happened with this species in these waters, because the pressure is not so high, and the algae maybe has changed some the habits. I believe that to continue the researchers in this field will be good for everybody. Is something news about algae population in the highlands. That is very, very interesting. In the same time, I want also to congratulate because after years and years, more than 45 years, I saw somebody from Ecuador. I want to say that in Galati city, where is a research institute, was also students from Ecuador in the Faculty of Food and Fisheries of the University of Galati. And it's a good time to remember that Ecuador has now very good specialists. Again, congratulations to you and to teams and to everybody who is joined to this hard work.
Thank you so much. Thank you for the invitation and greetings for Ecuador. I hope to see you someday.
We wish to.
Yes, why not to cooperate with research
It's
institute?
a pleasure.
It's an opening also for Professor Longo. We're waiting both of you to try to develop future research in algae, because it is very important. We notice in fish culture that when the population of Scenedesmus algae and also Chlorella algae, green algae, is very developed. We didn't have an impact of diseases on fishes. And maybe it's also very important for the earth pond system to growth fishes to try to develop green algae population, so what to avoid to use antibiotics in treatment of the diseases. We can develop a lot of discussion, but I will stop here. Best wishes from us to all team. Thank you very much.
Thank you again. Thank you.
Okay, so Carla make a lot of interesting point of view here and start debate. Okay, the next one will be presented by Ionica Bejenariu from Institute of Research and Development for Aquaculture, Ecology, Fishing and Aquaculture, Romania, Galati. And the title is Effect of Dietary Spirulina on the Growth and Health Status of Carp, Cyprinus carpius, Linarius 7058. So, Ionica, you have the...
Hello, my name is Bejenariu Ionica, and today I will present you the paper.
A little bit stronger, I don't hear you. Mai tare puțin.
My name is Bejenariu Ionica, and today I will present you the paper named Effect of Dietary Spirulina on the Growth and the Health Status of Carp Cyprinus carpio. In recent years, increased attention has been paid to dietary supplementation, such as probiotics, microalgae, and medicinal plants to improve growth performance, non-specific immune responses, and feed efficiency in aquatic animals, as they are inexpensive and easy to prepare. Spirulina plantensis is a microalgae recognized as an excellent dietary supplement due to its high content of protein, vitamins, trace minerals, and minerals such as potassium, calcium, iron, magnesium, and copper. For animals, including dogs, cat, and fish, spirulina offers important benefits. It supports immunity, skin and coat health, improves digestion, contributes to detoxification of the body, and intensifies the color of fish. It is administered in powder or tablet form, mixed with regular food. In previous studies, various beneficial effects have been demonstrated, particularly on growth performance, immunity, and disease resistance through dietary supplementation with spirulina plantensis in various fish species such as Siberian sturgeon, common carp, rainbow trout, and Nile tilapia. Knowing all this information about the positive influence of spirulina on different animals, we set out to investigate the impact of dietary supplementation with spirulina plantensis on the growth performance of carp and on its health status as assessed by the key hematological indicators and blood biochemical parameters. The experiment was carried out over a period of 40 days, and in order to evaluate the influence of different concentration of spirulina, a free experimental variant was organized. The first experimental variant, control variant, without spirulina addition, used as a reference point for evaluating the effects. The second experimental variant, with 4% spirulina powder added, designed to highlight the impact of moderate level of supplementation, and the last experimental variant with 6% spirulina powder added, designed to analyze the effect of a higher level of dietary supplement. Every experimental variant was compressed with 2 growth units, and in each growth unit, 10 fish were introduced. In first experimental variant, the average weight of fish was 87 grams per fish. In the second experimental variant, the average weight was 89.9 for each fish. And the last experimental variant, the average weight of fish was 89.9, exactly at the second variant. In order to the material biological was fed with food containing 34% protein, to which spirulina powder was added. Spirulina powder obtained from the pilot research and development station for algae cultivation was cultivated in modern photobioreactors operated in a fully controlled environment at the Institute of Research and Development for Aquatic Ecology, Fishing, and Aquaculture in Galati. To incorporate spirulina into feed, next step were followed. First step was to dissolve and homogenize spirulina powder in a 2% gelatin solution. The next step was to incorporate the result solution into the granulate feed. And the last step was to dry the pellets in an oven at 25-degree temperature for 2 hours. In the final, we obtain food with 4% spirulina added and 6% spirulina added. In order to determine the main biotechnological indicators at the beginning and the end of the experiment, the juvenile carp were weighed and measured, and the following parameters were calculated: specific growth rate, feed conversion ratio, and protein conversion ratio. To evaluate the influence of different concentration of spirulina on health status of juvenile carp, hemoglobin, hematocrit, and red blood cells count, and various biochemical blood parameters were analyzed. For this purpose, at the end of the experiment, biological blood samples were taken from 8 fish in each experimental variant, extracted randomly. The hematological indicators were determined by a method already known in specialized literature. Statistical analysis of the data was performed using Excel and SPSS statistic software. Single factor ANOVA and the t-test were used to compare the experimental group, and the results were considered statistically significant at a value of p lower than 0.05. Through the experimental period, the main water quality parameters were monitored, such as temperature, oxygen, every day, and compounds 2 times a week. The graphs show that both temperature and oxygen are within the optimal range for study species. The pH and nitrogen compounds were also maintained with the optimal range throughout the experiment. pH varied between 7 and 9. The nitrates were lower than 0.1 milligram per liter, and the nitrates vary between 15 and 23 milligram per liter. Regarding the lactate weight regression in all three variants, the control variant and the two experimental variant, one with 4% spirulina and the last with 6% spirulina, we can observe that the higher value was recorded in the variant with 4% spirulina added, indicating a very strong correlation between spirulina addition and growth. This upward trend shows that spirulina supplementation not only stimulates growth but also reduces individual variability, with 4% being the most effective level in generating a uniform and predictable growth response. In this table of biotechnological indicators of juvenile carp, we can observe that at the beginning of experiment, biomass in all three variants were almost the same, and at the end of the experiment, the highest biomass was recorded in variant 2 with 4% spirulina added. We also can see that all performance indicators, such as specific growth rate, food conversion ratio, and protein efficiency ratio, was recorded higher value in the variant where spirulina was added at a concentration of 4% without affecting survival. Hemoglobin variation, one of the most important hematological indicators, showed a statistical significant decrease in variant in which the feed was supplemented with 4% spirulina compared to the control variant. In the variant in which the supplementation was increased to 6% spirulina, the hemoglobin concentration was considerably closer to that of the control group. Overall, the results suggest that moderate spirulina supplementation can induce transient changes in the hematological profile, while a higher level seems to favor the stabilization of hemoglobin, possibly through a progressive adaptation of fish metabolism to an increased intake of functional nutrients. The second hematological indicators, hematocrit, shown an increase in value in the experimental variant with lower spirulina concentration, with a statistical difference compared to the control variant, and a decrease in the variant with 6% spirulina, also with a statistical difference. This variation may be associated with either changes in plasma volume or difference in erythrocyte size and volume, suggesting a distinct adaptive physiological response depending on the level of spirulina supplementation. The last hematological indicators, red blood cells, increased slightly in variant with 4% spirulina compared to the control level. In contrast, in the variant with 6% spirulina, the number of erythrocytes decreased, indicating a change opposite to the observed at the lower concentration, but without statistical significance between the three variants. The results suggest that moderate supplementation with spirulina, 4%, has a favorable effect of erythrocyte production, while higher doses may exceed the optimal physiological threshold, leading to a reduction in erythropoiesis. After we based on result obtained from the determination of hemoglobin, hematocrit, and red blood cells, the derived erythrocyte constant were calculated. So the first erythrocyte constant mean corpuscular volume showed an increase in the experimental variant with 4% spirulina compared to the control variant. This increase suggests that the erythrocyte formed in the presence of a moderate intakes of spirulina are larger, possible as a result of intensified erythropoiesis or accelerate erythrocyte maturation. In the variant with 6% spirulina, mean corpuscular volume was reduced, but with statistical significant difference like mean corpuscular volume in variant with 4% spirulina. The mean erythrocyte hemoglobin shown an inverse proportional evolution compared to the mean corpuscular volume in 4% spirulina variant, being lower in this variant, suggesting that although the erythrocytes are larger in this variant, they contain less hemoglobin per cell. The mean corpuscular hemoglobin, the last the erythrocyte constant, recorded the highest value in the variant with 6% spirulina, which is statistically significant compared to the variant in which 4% spirulina was used. This increase indicates a clear improvement in hemoglobin contents of erythrocytes, suggesting that despite the slightly reduced erythrocyte volume in this variant, the hemoglobin density inside the cell is significantly higher. From analyze the blood biochemical parameters, we can see that total protein, albumin, and globulin showed consistent changes, especially in the 6% spirulina variant, with statistical difference between the three variants. The significant increase in total protein and albumin confirm that spirulina, through in high contents of crude protein, essentially amino acid, and bioactive factors, can improve protein synthesis and metabolic efficiency in fish. Also, albumin globulin ratio decreased in 6% spirulina variant, mainly due to the marker increase in globulin. A low albumin globulin ratio is specific to organisms with increased immunological activity, indicating stimulation of defense process. Serum glucose decreased significantly in both spirulina variants, indicating a reducing in metabolic stress and a possible improvement in stress tolerance. Spirulina is known for its antioxidant content. Analysis of liver enzymes such as aspartate aminotransferase and alanine aminotransferase a clear and significant differentiation between variants. At 4% spirulina, the significant decrease in aspartate aminotransferase value indicates a hepatoprotective effect, possibly due to the antioxidants and anti-inflammatory compounds in spirulina. In contrast, in 6% spirulina, aspartate aminotransferase, significantly suggesting either metabolic overload or an adaptive response associated with an increased overall metabolic rate. It is important to note that alanine aminotransferase, a more specific marker for the liver, decreased in 6% variant, indicating good liver function despite the increase in aspartate aminotransferase. After we analyze the experimental base condition and evaluates the most important biotechnological indicators, hematological indicators and blood biochemic parameters, we can draw the following conclusion. The infrastructure in which the experiment was organized ensured, thanks to advanced water quality control technique, normal condition for the development of crop biomass in all three experimental variants, which led scientific credibility to the result obtained. Overall, the analysis of length-weight regression demonstrates that supplementing feed with spirulina has a beneficial effect on growth uniformity in fish. The 4% spirulina level proves to be the most effective in generating a predictable and uniform response, while the higher level of spirulina continues to deliver many good results, confirming the favorable role of spirulina in fish nutrition. Analysis of key biotechnological indicator highlight that spirulina is an effective and safe protein additive, and 4% of spirulina is optimal dose for maximizing growth, protein utilization, and feed conversion, with no adverse effect on fish survival, being optimal in terms of cost-benefit ratio. The main conclusion resulting from the analysis of hematological indicators is that supplementation with 4% spirulina appears to promote erythropoiesis stimulation. This is reflected in an increase in hematocrit and red blood cells count, indicators that suggest increased activity of hematopoietic tissue. However, this stimulation is accompanied by a reduction in average hemoglobin content of red blood cells, indicating that although more red blood cells are being produced, they may be less loaded with hemoglobin. This situation may represent a transitional stage of physiological adaptation in which the body has not yet managed to achieve an optimal balance between the process of red blood cell production and hemoglobin synthesis. The evaluation of blood biochemical indices shows that the addition of 6% spirulina is the option with the best metabolic and the immunological benefits, but it can induce intense physiological activity, as observed by the increase in AHT. However, the reduce of alanine aminotransferase value suggests that this dose is well tolerated by the liver. Thank you for your attention.
Have you also measured oxidative and inflammatory markers? This is the question.
No.
Because, okay, I can, very interesting presentation. Thank you. What is your idea about shelf life of fish? I think my idea is that you can, if you put spirulina in feed, you can improve the shelf life, the stability of fish. For this, it can be interesting also to study, maybe to measure some inflammatory and oxidative markers. But my idea is that you can improve the shelf life of product.
Yes.
What do you think?
It could be. It would be a next research for us. I don't understand.
I repeat. Can you hear me?
Ionica, poți să-mi spui mie și îți spun eu, domnul profesor.
Dar nu am înțeles ce vrea, am răspuns, nu am înțeles ce vrea domnul profesor.
Să-i răspunzi încă o dată la dacă în viitor pentru hrănirea peștilor folosești spirulină.
Yes, in the future we want to use spirulina in fish food, and we'll do more research about this.
Okay.
Thank you.
Okay, thank you. Thank you.
Sorry, I want to say that maybe, Mr. Professor, and with excellent team from Galati Institute, you can do in near future a research project regarding the use of spirulina for aquaculture. What do you think? Maybe it's an open gate for a new research project.
Yes, it's a new idea for a new improved food for aquaculture, and this is why we want to do more research about this added spirulina in a different concentration.
I think it's very important, this symposium, not only for the subject that you are presenting, but also for the fact that you discover new ways for collaboration. And I congratulate all for this benefit. I'm very happy that I am present and here in this symposium. Thank you very much.
Thank you too.
Thank you. So the next presentation will be valorization of Posidonia oceanica, residue and sustainability growing media, presented by Francesca Vanucchi, Cristina Macchi, Serena Doni, Grazia Mascandaro, and Eleonora Peruzzi. But first, Nicole Petculescu will make a short presentation of Francesca Vanucchi.
Ciao Grazia, ti sto vedendo, ben arrivata, siamo contenti di averti con noi. So hello Grazia Mascandaro, she is together with us, together with also her team Francesca Vanucchi, both they are researcher at the CNR, Consiglio Nazionale di Ricerca di Pisa. Grazia Mascandaro is research director, and her research subjects are in soil science, enzyme activities, phytoremediation, humic substances, bioremediation, and organic matter. And regarding Dottoressa Francesca Vanucchi, her research focused on agri-industrial waste recovery through the application of biological technologies, composting, vermicomposting, and their valorization on growing media for agricultural purposes within a circular economy framework. So please, Grazia Mascandaro, Francesca Vanucchi, Cristina Macchi, Serena Doni, you have the floor.
Thank you so much. Good morning, everyone. Today, I'm showing you the research activities that we carry out about the valorization of Posidonia oceanica residues. We started from two environmental issues. The first one concerned the extraction and the use of peat in agriculture, which leads to increased carbon emissions and habitat loss. And since peat is not a renewable resource, it is essential to find sustainable alternatives. At the same time, there are challenges related to the disposal of degraded sediments and Posidonia residues. In fact, large amounts of sediments are collected during the cleaning of ports and waterways to ensure the navigation. And these sediments are often contaminated by heavy metal and organic pollutants. In addition, the rising of sea temperatures and eutrophication promote the proliferation of Posidonia, generating large amounts of residues that accumulate on the beaches, and these residues must be removed to maintain beach clean for the tourism. So to address these issues, we tested biological technology for the recovery and the valorization of these wastes. Specifically, we implemented a composting process in which sediments and Posidonia residues were mixed to produce a substrate suitable for agricultural use in compliance with European and national regulation. The first step concerns the composting process, and Posidonia residues were collected in Rosignano Marittimo, a location on Tuscany coast, and these residues were washed 4 times to reduce the salinity, which could negatively affect the composting process, and then chopped to reduce the particle size. The composting piles were prepared with different proportions of Posidonia, green waste, and sediments, and in particular, the sediments had previously been decontaminated by phytoremediation and land farming, while the green wastes were collected by the municipality. The composting process lasted 6 months, and the pile were periodically turned to ensure the aerobic decomposition of organic compound by microorganisms. And in addition, several physical, chemical, toxicological, and biochemical analyses were carried out to monitor the efficiency of the process. The best results were obtained with the mixture contain 20% of Posidonia and 80% of green waste. And here we presented the most significant findings that demonstrated the efficiency of the process. In particular, temperature measured in each pile during the process reflected the degradation dynamic. And in particular, during the first 120 days, we observed an increase in temperature with peaks following the turning of piles. And in this phase, the more labile organic compounds were degraded by microorganisms, leading so a temperature increase. However, the temperature remained above 55 degree for 14 days as required by the Italian regulation for the sterilization of the material. And the subsequent temperature decrease indicate the maturation phase, characterized by the formation of stable organic matter and the reduction in the carbon and nitrogen ratio, confirming the stability of the final product. And the efficiency of the process were also confirmed by the microbial analysis, which follow the same trend as other parameters. The compost composed of Posidonia and green waste also fully met the threshold defined by the Italian and European legislations, so it can be used as a component of growing media for agricultural purposes. So the suitability of compost as a component of growing media was tested in an experiment carried out in a nursery. And specifically, we used a randomized block design to test 3 different growing media containing varying percentages of Posidonia compost and decontaminated sediment. These growing media were compared to peat as the control. The trial involved the cultivation of common ornamental plants over to vegetative cycle. We performed analysis on both the growing media and the plant performance before the starting of the experiment, during the plant growth, and at the end of the experiment. For example, for the growing media, we assessed the nutritional status by quantifying nutrient content, and we evaluated the stability through the enzyme activities. In addition, ecophysiological analysis such as leaf gas exchanges, chlorophyll content, and antioxidant activities allow to monitor the plant health status during the experiment. The initial analysis of each growing media confirmed their compliance with Italian regulation for both mixed and base plant growing media with very low heavy metal content. And in addition, all the growing media showed the nutrient ratios within an ideal and acceptable range for the plant growth. And in particular, the calcium, magnesium, and potassium ratios were higher in the Posidonia-based substrate than in peat. And this optimal condition persisted during the plant growth period, suggesting a long-term nutrient availability for evergreen plants and reducing the need of additional fertilization. Concerning the plant performance, plant grow on substrates containing Posidonia compost and sediment showed a similar health status compared to the control in terms of plant growth, pigment content, and gas exchanges. However, the enhanced nutrient status in the Posidonia and sediment growing media reduced the antioxidant activities observed in the control, suggesting so a lower plant stress and better performance. So in conclusions, the research activities demonstrate the efficiency of composting in recovery and valorization of Posidonia residues, creating a substrate suitable for agricultural application in line with the legislation. In particular, the use of Posidonia compost as component of growing media together with the contaminated sediments was a sustainable solution to reduce the use of peat and the chemical fertilizer in horticulture. So thank you for your attention.
Thank you. If someone have question?
I also want to congratulate Francesca and Graziana and all the research team from Consiglio Nazionale di Ricerca for their work, for their scientific activity during the time. They are very well known for very good results in research practice within the international scientific world. And I think that it's a very good subject for the agriculture and for everything that it is related with agriculture regarding this paper. Thank you very much.
Thank you for inviting us.
From Waste and Sustainable Vineyard Resource, the presentation will keep by Mihai Tudor from Station of Research and Development of Viticulture and Winemaking, Târgu Bujor. Mihai, you are online?
Yes, thank you very much for your invitation, and I'm glad to be part of this very special scientific event. Our presentation that includes also our host, Mr. Mihai Vintila, which we have a close relationship with the Institute of Aquaculture. Let me share the screen. Okay, let's share it. Okay, is it okay? Can you see
now that yes.
Okay, so I'll do it in full screen. In slideshow mode. Let me see. Is it okay?
Yeah, it's okay. You can start the presentation.
Great. So here our research presents some general aspects regarding the use of algae and towards viticulture and its application in viticulture. But first of all, I researched some aspects regarding our current trends, our current economical trends, And of course, there is a flawed relationship between humans and the natural habitats, having negative consequences such as biodiversity loss and a never before seen global crisis. When looking at the environmental aspects, we are all aware that there is significant climate change, air pollution, water scarcity and pollution of the water sources, deforestation, loss of biodiversity, and waste accumulation. Unfortunately, our current linear economic systems, which allowed our societies to prosper and all our capital forms are based on non-sustainable resources. And that's why when looking at renewable versus non-renewable resources, we can also see that renewable resources are can easily be replenished, and versus non-renewable resources that take millions of years to form and cannot be regenerated within a human life frame. That's why we need to think about our agricultural sustainability and transitioning from our current linear economy to a circular economy, and of course think about bioeconomy and reduce our dependency on new natural resources extraction and increasing the resources for alternative cycles, and also develop, when talking about bioeconomy, we also need to minimize the impact of human activities on natural environments and maximize human health and natural resources. When talking about circular economy and bioeconomy, the concept of intersection between these two concepts is circular bioeconomy, which currently is creating confusion and difficulty in the scientific world, because it's a framework that the European Commission is defining as a framework that reduces the dependence on natural resources and transforms manufacturing, promotes sustainability production of renewable resources, and promotes their conversion into various bio-based products and bioenergy while growing new jobs and industries. Our aim was to review the main techniques that use algae to enhance soil properties and generally the health of the vineyard plantation, respecting the principles of circular bioeconomy. Of course, we researched all the main scientific databases using a timeline between 2013 and 2025. When talking about the sustainability aspects of the vineyard plantation, this ecosystem is endangered because of soil function loss due to traditional agricultural practices that are repeated over time, and especially in the European region of the vineyards, and also and especially the Balkan region, we can talk about the loss of organic matter in the soils and extreme erosion that is caused by runoffs due to the fact that the amount of precipitations Although when talking about when consulting a certain timeline, we can see that average precipitation values reach the specific values for the zone, for the viticultural area. Unfortunately, climate change has modified the frequency of precipitations with long periods of drought and followed by extreme weather phenomena that cause runoffs. So algae can become a sustainable alternative that allow preserving our viticulture sector. This for the authorities, for the managing authorities, can be an environmental problem due to the fact that this algae as an organic matter source are costly to remove from the beaches, and that's why this can become a very good alternative, a circular, biocircular alternative for sustaining our vineyards, due to the fact that many vineyards are placed along the coastlines. Here in Bujori, it's not the case, but it is a good alternative. So, as soil amendment tools, the use of algae can increase crop biomass, improving yield and fruit quality, and resistance to abiotic and biotic stressors. They also improve water and nutrient retention by delivering bioactive substances. And they also can increase the concentrations of growth regulators and organic compounds in the soil medium. When thinking about the presence of essential macro and microelements, studies show that nitrogen content reached or had noticeable improvements when composted seaweed was used, and also can moderately enhance soil nitrogen storage. Together with amino acids, hormones, and fatty acids, nitrogen, phosphorus, and potassium can provide a healthy plant growth or a healthy vine plantation, resulting in significant nutrient content increases. When thinking about the use of algae, we can talk about maybe additional heavy metal presence. When studying the literature, we saw that when healthy source of algae was used with low concentrations of heavy metals, the resulting residual concentrations present in the soil were below limits established by the EU legislation. A special case was mentioned for cadmium and nickel, which had a slower turnover rate. But as an idea, or as to implement this kind of strategies here in our vineyard plantations, I think that close monitoring with analytical methods such as ICP-MS would be a great tool to screen out possible problems with high concentrations of heavy metals. And also there is another way to remove or change the to create some macromolecular complexes using mixes that that use algae and yeasts that result from vine fermentations, because when consulting the literature, I saw there are some studies that have positive results in the use of yeasts that resulted from the vinification process in complexing heavy metals present in algae composting. From an economic perspective, here at our research station, we are already using grape pomace in a composting experiment to enhance soil characteristics, but we can all say that, or we all can agree that composting process can lead to significant reduction in costs, and for the agricultural sector, including the viticulture, composter presents a cost-effective and sustainable substitute, with a potential of lowering input costs that represent maybe the use of fertilizers, inorganic fertilizers, and so on. Here I mentioned the main case reports regarding the economic perspectives. They all mention local communities in the US. These were the most representative, with important job generation opportunities. For example, in the Iowa Department of Natural Resources, that saved nearly $100 million and created over 19,000 jobs, using these type of strategies of circular economy and bioeconomy. In conclusion, our current review highlights the potential of seaweed and that can maintain soil fertility for multi-annual crops. The use of seaweed compost was responsible for carbon-nitrogen ratio amendments, and this eco-friendly management of marine byproducts remains essential in the context of climate change, helping to reduce the reliance on traditional mineral or oil-derived fertilizers, and achieving, of course, the EU legislation reduction target of 10% by 2030. Thank you very much for your attention. If there are any questions.
Apologies. So, as I was saying, my name is Bianca Mata, a research assistant and marine biologist at the National Marine Research Institute Gheorghe Ionescu here on the beautiful Romanian coast. And today I will be presenting our team efforts. Just to mention, the lead researcher for this project is Dr. Oana Alina Marin, our macroalgal expert, and I am supporting her in this project and design experiments. Next slide. The presentation will go through a detailed section about the project, and I will talk about the two macroalgal species that we are currently conducting research on, Ulva and Cistozeira, and finally some conclusions about our progress so far. Ex-Aqua is a 3 years European-funded project aimed at boosting scientific research in algae aquaculture, micro and macro, with a vision to make the National Institute for Marine Research and Development, Gheorghe Ionescu, or NIMRD, a center of excellence in algae farming, supporting innovation, climate change, local economic development. And this presentation introduces the project's primary objectives and summarizes the progress to date, aiming to promote sustainable algae aquaculture across the Black Sea region. Here you can see all our partners of the consortium. First of all is us. You might know or not, but we've updated our logo in the meantime, and hopefully we'll update them across all our communications channels. Our technical partners from Ghent University in Belgium, AVI in Germany, the Black Sea Advisory Council, Lomartov from Spain, and CMMI from Cyprus. The project follows 4 key pillars in order to achieve its mission, and the mission aligns with the EU's blue growth economy, including protection of the marine environment and sustainable development, but also fostering new jobs and market opportunities. The action plan for the next 3 years ensures the project team is on track with their progress and deliverables, working towards funding, policy, and regulatory mechanisms, research priorities, and strategic goals for long-term outcomes in the next 10 years using a strategic roadmap. Also, through workshops, summer schools, and other activities that foster partnerships in science and knowledge exchange, the project aims to create a cluster on applied marine aquaculture in Romania. So why do we cultivate macroalgae? Well, it's been well proven across the globe that they are beneficial for several industries, from pharmaceutical to biofuels, and that their biocompounds can replace or enhance several products for human and environmental benefits. But we do have a bit of a hard work to conduct as well, changing people's perception about algae in Romania being perceived as bad aesthetics, especially for tourism. So the project does go a bit into the social aspects of blue economy acceptance as well. And for that, we conduct testing and application cultivation techniques on 2 local algae. Uva, the green one in the picture above, you can see there. It's a green algae, green macroalgae, less pretentious, but common across Europe and worldwide. And the brown alga Cystosera barbata, the picture below. It's a critically endangered brown alga, common in the Black Sea, and an important key biodiversity supporting species. We cultivate uva for its high nutritional value and bioactive compounds, and Cystosera for ecological reconstruction activities only. Both species undergo cultivation techniques adapted to Black Sea specific conditions. Here you can see a picture of our new laboratory containing our babies, just as I like calling them, and our upscaling efforts so far. On the left side here, you can see the green algae in containers, and on the right side, the brown algae, just on the far right side, grown on different types of substrates. I will go in more details about each algae development in the following slides. Firstly, uva. The green algae are cultivated mainly in flasks and large containers, but as you will see later in the slides, we've also done some experiments on ropes as well. In the pictures here, you can see a bit of the process we undertake to cultivate uva, and I will run you through a bit. So first, we collect the material from the natural environment. We induce correlation in the lab, after which we leave them in the hatchery area, mimicking natural environmental conditions specific to the species, and then we wait for it to develop the juveniles in the nursery. From this, we scale up further, meaning we move them in flasks suitable for their size, and from there we scale up more, monitor and measure relative growth rate. Obviously, it's an agile process. We adapt and change as required, and we learn as we go with the experiments. Here are some more details about uva. These experiments, because we are in our first year of experimental laboratory cultivation trials of the project, they are grown and monitored since January this year. We received support and recommendations from Ghent University in Belgium for developing effective cultivation methods for Black Sea salinity conditions. We used only F2 nutrient formula, culture medium popular in microalgae cultivations. And our measurements show the relative growth rate between 0.8 and 6% per day. With hard substrate testing for this species was done on both synthetic polypropylene and natural wood rods. And already we achieved our main goal with this species of reaching 200 liters of cultivated algae in laboratory settings, meaning that we moved from technology readiness level TRL 2 to 4. Moving forward to Cystosera. Being a more sensitive species, both in environmental impacts but reproduction potential ex situ as well, we first carefully designed the experiment research plan. So we decided to do 7 replicates for each of the 2 different hardness of substrates. Similarly to uva, we collected a donor reproductive material from the natural environment, where the species is still found in patches across the Romanian coast. We followed a rigorous process for handling the samples, ultimately waiting for it to attach to the substrate and start growing. I won't lie, it was a lot of tension not knowing what will happen, considering how sensitive this species is. Well, luckily, Sister Zira was friendly with us and appreciated how well we treated her, and so she accepted her new environment and started growing. All tested substrates were successful in supporting Sister Zira's development, with obviously some differences in relative growth rate, with hard substrates such as travertine showing the fastest talus elongation. Here are some more pictures of our Cystosera babies on the hard substrates. In the pictures, you can see different growth rates, stages, mainly on travertine, and the orange ones are ceramics. And I will hold on a bit because I think I'm talking too fast. So you can admire a bit the pictures. Just on the first picture of ceramics, you can see the very microscopic bits that Cystosera left on the hard substrate and how much of a difference is now in, I think there's 6 months now since we first started with the Cystosera. Here again is a comparison with the softer substrates on ropes, which show the slower growth rate, but with polypropylene being the winner, where the thallus reached 3 centimeters. And some more pictures of the rope experiments here. Similarly, example of the different growth stages and pictures of both the synthetic and natural growths. And just to mention, our aim is to take these experiments out in the sea, actually, and monitor their adaptation capabilities for ecological reconstruction. So as a first thought, we believe hard substrates will likely be more successful than the ropes due to mimicking the species' natural substrate references much better. To finish, as a comparison between vulva and Cystosera, our relative growth rate statistics show that vulva grows the fastest, but this is expected as Cystosera is a slow-growing species, and our main focus is to obtain biological material for ecological restoration, so we are a bit more careful with it. And marine algaculture is currently absent in Romania, but despite of the absence of a national tradition, in algaculture, the project is committed to advancing its development and aligning our practices with established European aquaculture standards. Our aim is to develop standardized methods for growing local algae, while also leaving room for adaptation and flexibility. And the role of the Romanian Marine Research Institute is to transfer these methods to the stakeholders and to support the future large-scale cultivation of macroalgal species and on the Romanian coast, enhanced by expertise of our partners. And the last slide. This is us. I'm on the right and my colleague on the left. Thank you very much for your attention and for inviting us to speak to you today. I'm happy to take over any questions. I'll drop my email in the chat and hopefully we can make some beautiful collaborations further. Thank you.
Okay. Stop, please, the share.
Yes.
Okay. First, I have a question. I was surprised to see how grow on travertine and travertine tiles.
It mimics the best the rocks, reefs out in the sea. So we've tried different substrates from travertine to shale to granite to ceramics, and travertine was the most successful one. And ropes a bit, but ulva grows better on ropes because Cystosera has a more limited base, then uva also floats in the water. So you can do whatever you want with it. While Cystosera is a bit more restricted due to the base that it has to attach, and yeah, just different biological aspects of the species.
Okay, thank you. If there are other questions, if not, we'll go to Research and Development Station for Fishery Nucet. They will present a paper, Research and Optimizing Method for Combating Cyanobacteria in Fish Ponds.
Good afternoon, ladies and gentlemen. My name is Marica Nino, and I will present the scientific paper entitled Research on Optimizing Methods for Combating Cyanobacteria in Fish Ponds. Cyanobacterial blooms are a major problem for aquatic ecosystem and fish farming activities, affecting water quality and the health of organisms. This paper compares the effectiveness of free control methods applied in experimental fish ponds, chemical treatment with copper sulfate, treatment with hydrogen peroxide, and the ecological method based on the use of barley straw. Over a period of 8 weeks, the physical and chemical indicators of the water, the concentration of chlorophyll A, and the density of cyanobacteria were analyzed in 4 ponds with a history of algal blooms. The experiment was conducted at the Research and Development Station for Fisheries Nucet in 2025 between June and August. 4 fish ponds with a history of cyanobacterial blooms were used. The treatments applied were B1 control with no treatment, B2 chemical treatment with copper sulfate, 0.4 milligrams per liter, weekly application, solution dissolved and dispersed on the surface of the pond, B3 treatment with hydrogen peroxide, 5 milligrams per liter, applied every 2 months, and B4 ecological treatment with barley straw, 50 grams per square meter. In this picture, you can observe the 4 experimental ponds. pH values showed significant variations depending on the treatment and the experimental period. In the control basin, B1, the pH decreased steadily, reflecting the accumulation of organic substances and the intensification of microbial respiration processes. In the copper sulfate treated basin, pH values ranged from 7.55 to 6.80. In contrast, the ecotechnological treated basins maintained better chemical stability. Dissolved oxygen showed a trend directly related to photosynthesis intensity and algal bloom severity. In B1, values ranged between 5.2 and 6.8 milligrams per liter, with a downward trend in recent weeks, indicating trophic stress. In B2, treatment with copper sulfate initially caused an increase, followed by a rapid decrease due to the decomposition of dead biomass. In B3, oxygen remained constant, and in B3, it gradually increased, indicating moderate photosynthetic activity and a stable redox balance. Comparatively, the average dissolved oxygen values were 20-25% higher in the ecotechnological treated basins than in the chemical treated ones. Water transparency determined using the Secchi disk varied inversely with the cyanobacterial density and nutrient concentration. In B1, transparency gradually decreased from 42 centimeters to 25 centimeters as algal growth intensified. In B2, after the application of copper sulfate, transparency increases sharply to 60 centimeters, but subsequently decreased to 35, indicating the reappearance of phytoplankton after the reactivation of nutrients released by lysis. In B3, values remained high, and in B4, they stabilized between 70 and 80 centimeters, demonstrating significantly higher water clarity in ecotechnological treated basins. Evolution of ammonium concentration reflected the intensity of mineralization processes and the efficiency of the treatment applied. In B1, the values increased steadily, indicating the accumulation of organic degradation products. In B2, there was a sharp increase to 1.85 milligrams per liter, followed by a slow decrease associated with subsequent nitrification processes. In B3, ammonium decreased steadily due to oxidation under the action of hydrogen peroxide, and in B4 decreased slowly but steadily. The average reduction in ammonium was 75% in B3, 55% in B4, and only 25% in B2. Phosphorus showed a similar trend to ammonium. In B1, levels increased from 0.18 to 0.41 milligrams per liter, and in B2, they peak at 0.45 milligrams per liter in week 3, followed by a gradual decline. In B3 and B4, values decreased steadily to 0.06 milligrams per liter, confirming the role of ecotechnologies in reducing phosphorus availability. Evolution of cyanobacterial biomass. Hydrobiological determination showed consistent variations in phytoplankton biomass. During the experimental period, significant variations in biomass were observed between the four basins. In the control basin, biomass increased steadily, reflecting progressive eutrophication and favorable conditions for bloom formation. In the copper sulfate treated basin, an initial reduction was followed by a partial recovery, indicating temporary chemical suppression and subsequent recolonization after nutrient reactivation. In the hydrogen peroxide treated basin, biomass decreased sharply, confirming strong oxidative inhibition of cyanobacteria without secondary pollution. In the basin B4 showed a gradual decline consistent with the slow release of phenolic compounds that inhibit algal photosynthesis. In this chart, you can observe the evolution of cyanobacterial biomass in the basin analyzed during the experimental period. Evolution of chlorophyll A concentration. The experimental data revealed clear differences depending on the treatment. B1 showed a continuous increase of chlorophyll A, indicating progressive eutrophication and dense proliferation of cyanobacteria. B2 showed a transient reduction followed by pigment recovery, reconfirming the short-term impact of chemical control. B3 showed a sharp decline, highlighting a sustained inhibition of pigment synthesis in cellular activity. B4 showed a moderate, steady decrease linked to the gradual release of natural algistatic substances. In conclusion, the research results confirm the effectiveness of ecotechnological treating methods in controlling cyanobacteria and stabilizing aquatic fish ecosystems. treatment with hydrogen peroxide ensured a 70 to 80% reduction in ammonia and phosphate compounds without ecotoxic impact, while barley straw provided a slow but stable effect. The copper sulfate-based chemical method caused strong fluctuation in parameters and secondary nutrient release, which limits its long-term applicability. Ecotechnological treating methods are a priority for the sustainable management of aquaculture's water affected by cyanobacterial blooms. Thank you very much for your attention.
Thank you very much. If someone have questions? No. If not, we'll go to the next one. Influence of nitrogen concentration on biomass accumulation in spirulina cultivated in photobioreactor. Collective is from Institute of Research and Development for Aquatic Ecology, Fishing and Aquaculture, Galati. The presentation will be kept by Alina Nicoleta Dobre. Alina?
Hello. Hello.
Hello.
I'm responsible for operating the Algae pilot, which is an innovative and unique project in Romania. Before detailing the subject of presentation, I give you a quick introduction and show you the main installation are configured and how they work. Next, please. Why this project? We are looking at the need for more sustainable protein source, the untapped potential of aquaculture in Romania, and the opportunity to create models that could be replicated across the country. What do you do specifically? We try out different ways of growing spirulina, see how well it performs both biologically and economically, and share our finding with university and the industry. What do you get? At the pilot station, we have a working model in place. We use a consist method for growing algae, and we expanding our research and training capability. Long-term impact, our work focus on promoting sustainable development in agriculture, finding ways to really less on traditional protein source, collaborating international and sharing our technology and foresting innovation through interdisciplinary education. Our station is organized into 4 areas, all centered around an advanced photobioreactor system used to cultivate Spirulina platensis. We three photobioreactor, each with a different capacity, support the main stage of the process, from research and development to micro production and full-scale production. We have initiation zone. We have a small photobioreactor with 3-liter chambers. Is used to try out cultivation method and check the quality of the biomass. The second zone is inoculation. We have a photobioreactor 20-liter unit. Take this result and produce small batch of spirulina. And finally, in the production zone, a large 200-liter tubular photobioreactors allow spirulina to grow in controlled way, sustainable for both continued and batch production. All the biomass we transferring and processing zone, where is driving with 14 degree. All cultivation process are overseen by dedicated software platform that provide an intuitive interface, real-time data visualization, and remote access. Critical parameters including pH, oxygen pressure, turbidity, temperature, and lighting are constantly monitoring to maintain optimal condition for culture growth. And because the operation of the station directly influences what comes next, it's time to focus on the subject of my presentation. As you know, spirulina is a species valued for its high content of protein, vitamins, minerals, amino acid, and essential fatty acid. Its high nutritional value. This microalga is cultivated on an industrial scale, being used in the manufacture of food supplement, animal feed, and various pharmaceutical products. The efficient production of spirulina biomass relies heavily on environmental conditions such as nutrient availability, temperature, pH, and light intensity. This factor play a crucial role in determining both the growth rate and the biochemical makeup of the biomass, as they directly influence cellular metabolism and the processes responsible for the accumulation of essential compounds. The objective of this research is to explore how different nitrogen concentration affect the growth and biomass production of Arthrospira platensis cultivated in Zaruc medium under controlled photobioreactor conditions. Nitrogen plays a key role in spirulina metabolism and development, as it directly supports the formation of protein, photosynthetic pigment, and overall biomass. In this context, the study focused on optimizing cultivation condition and photobioreactor to achieve the highest possible yield, comparing how spirulina performs at various nitrogen level, and ultimately identified the nitrogen concentration that offer the best balance between economic and ecological efficiency. The methodology we chose use a small photobioreactor with 2 chambers, PBR, volume of each chamber 3 liters. We use free lever or off nitrogen suppleed or as nitrate sodium with nitrogen limited with 0.1 gram per liter, nitrogen optimal 20.5 gram per liter, and nitrogen enriched 5.0 gram per liter. We have a triplicate for treatment. The experiment was run in several separate, so that each treatment was tested at least 3 times in total. Duration, 12-day culture period. We use standard medium Zaruc, inoculum with 10%. The other parameters remained unchanged. Temperature 40 degrees, light 400 lux, and pH 9 with automatic aeration. Result and discussion. Growth performance was monitoring daily to determine the influence of nitrogen source to the different culture condition on the productivity of Spirulina platensis, optical density, and dried biomass of each culture. We monitoring every 2 days. The ANOVA test applied for each day of the experiment showed statistically significant difference between of free nitrogen condition on all 6-day analysis. As you can see in the picture, during the 12 days of cultivation, the color of the culture changed visibly. At the beginning, the sample had a light green color, showing the cell density was still low, As the culture grew, the color became gradually darker and shifted to a strong blue-green. This visible change clearly showed the increase in biomass and pigment production. This is the evolution and nitrogen enriched spirulina biomass. And the next slide, you can see the nitrogen limited culture. The trend was reversed. In the first few days, the culture actually appeared darker and more intense, but as the nitrogen was quickly used up, the color slowly faded. This decrease in intensity indicates lower pigment production and lower biomass growth caused by nitrogen stress. We observe in the evolution of Spirulina platensis biomass the medium with optimal nitrogen level show you steady growth and stable value indicate balanced photosynthesis and healthy biomass. Under nitrogen limitation, the culture grew quickly at first but showed a sharp decline after day 3 due to the nitrogen decrease, reduced protein synthesis, and lowered photosynthetic activity. In the nitrogen-rich medium, growth was initially slow but continued to rise, reaching the highest OD values by the end, supported by rich nitrogen and sustained photosynthesis. And the evolution of dry biomasses and nitrogen optimum constant grow until around day 10, then a slight decline. The medium has a balance with nitrogen concentration, indicating support stable and healthy growth. For nitrogen enriched, accelerated growth until day 10, a slight decline. Excess nitrogen stimulate protein synthesis and biomass accumulation. And finally, for the nitrogen limited, we see rapid growth until day 3, then a steady decline. The cell use up the initial nitrogen reserve, but then enter nutritional stress, indicating protein synthesis and pigmentation decrease. For the specific growth rate, nitrogen optimum moderate distribution with a decent average, relatively low variability, indicated stable growth. For the nitrogen limited, lowest average, high variability and stable crop, indicated nutritional stress and poor performance. And the nitrogen enriched, highest average growth rate, tight distribution, efficient and predictable crop indicate good adaptation to excess nitrogen. After calculation average productivity, we found that nitrogen limited culture showed very low growth. The optimal nitrogen culture grew steadily and well, and the nitrogen enriched culture reached the highest growth but needed careful control to avoid the stress. And about the doubling time, we observe for nitrogen limited only present between days 1 and 3 with a doubling time of 4.5 days. Spirulina grows relatively quickly in this phase, even if nitrogen is limited. For nitrogen optimal, initially day 1 and 3, spirulina has a moderate doubling time, indicated good growth. On day 3-5, the doubling time increases, so growth slows down. On day 5-7, the time decrease, signaling an acceleration of growth. On day 7-10, the doubling time increases, suggesting stagnation. For nitrogen enriched during day 1-3, very high doubling time, indicating slow growth. From day 3-5, doubling time remain high, indicating stagnation. Between 5-7, time decrease, accelerated growth. By day 7-10, time decrease even more, good, indicating good growth. One moment, please. What is the optimal day to harvest spirulina? The best harvest time is around day 10 under optimal nitrogen. Will nitrogen limited culture peak early at day 3, and nitrogen enriched culture reach their maximum around day 12. And now for conclusion, dry biomass accumulation was highest and nitrogen-rich culture, confirming superior metabolic efficiency. For specific growth rate, peaked under nitrogen-enriched, reflecting cell division and nutrient assimilation. Doubling time was shortest in nitrogen-rich and longest and nitrogen-limited, highlighting the impact of nitrogen availability on growth kinetics. For OD680, increased significantly under nitrogen-rich conditions, indicated enhanced photosynthetic activity and cell density. Day 10 under nitrogen optimum condition is the ideal point for harvesting, offering the best compromise between quantity and quality. The average productivity of spirulina is directly proportional to the nitrogen concentration. Overall conclusion, the nitrogen availability has a major impact of the performance of spirulina culture. The optimal nitrogen regime represents the most balanced option, economically ecological. Thank you for your attention. I will the spirulina is a powerful superfood for better health. If you have any questions, feel free to ask. Thank you.
Thank you. Okay, if are questions now. Okay, this was the last presentation, unfortunately, because it was interesting. And now we can debate if we have something to debate. But first, I want to thank you for being with us, a special Professor Vincenzo Longo, Carla Sandovan-Guano, who can make a lot of stress to be in the middle of the night to present and be with us. Francesca Vanucchi and Grazia Mascandaro, who was also online. The friend from Egypt, Professor Darwish, Professor Hassoub. We are open to more cooperation, and I hope the internet will work better next time, because the presentation was really interesting. The friend from
Thank you very much.
Okay.
Thank you very much, and hope to see you again.
Okay. The friend from our Academy of Agriculture and Forestry Science of Romania, the head of the Academy, Mr. Ioan Jelev, the Vice President, Aurel Badiu, and the Secretary of the Food Section Industry, who was close to us to make this good event. And also we want to thank you to all from other institute and also research center who present the information in most varied way. Important, the long part of the presentation will be published in our magazine, who will be published in end of December this year. And I hope there you can get more information and also contact information if you want to share the information. If are not other consideration, Mr. Petculescu?
Yes.
You have?
Of course. Of course.
Okay.
I'm very happy. I'm really, it was a regal scientific event. I would say it was an honor to see so many specialists from all over the world, exactly from all over the world, because we flew from Italy to Egypt, to Ecuador, I know that also the professor from Costa Rica, he was online. So, and finally to Galati and to Bucharest, where we are. Very interesting debate, very good topics. And I think these are the major steps in order to run for a new scientific international projects on short and medium term. And our section of food industry, it's very honored to be present and very proud of you, dear Institute and dear Director, Mr. Floricel Maricel Dima. And also thanks to Michael, because he was the moderator and the soul of this event, and also to the scientific director and the scientific soul of this event, Mr. Neculai Patriche. I'm very proud to say that he is a correspondent member of our food section. And all of your team, you are fantastic. And me, as scientific secretary, I'm very proud of you, very keen. Thank you very much. Thank you, Professor Vincenzo Longo. I told you that will be a very interesting event, and I think I was right. And thank you, Graziana. Thank you, Francesca. Thank you, Carla. Thank you all. Thank you, dear colleagues from all the institute, from Nucet station, from Bujoru scientific station, from Grigore Antipa Institute. Thank you to all.
Okay, Mr. Longo, nothing to tell to Eld?
Great. Okay, I think that it was very interesting today to know, to hear several presentation, and I think that we have different expertise, and we can, as Nicole suggest, to find a call and to present a project, to submit a project together. And today it was very important to know each other, and maybe we can try to submit some project, to write some project together now. Because there are different, we are involved in different field of algae, and maybe we can put together and to see the importance of this algae. Thank you very much.
Thank you also. We are a unity in diversity. Mahatma Gandhi says something like that. So I think we are exactly, but with research the head. Domnul Jelev, dacă aveți o intervenție finală, vă rugăm. Microfonul, domnul Jelev. Dă tu microfonul.
Only a very short intervention. I was very happy to have this opportunity to take part of such an important international symposium with such many very interesting papers and such a large international participation. And congratulations for you and for General Director Dima for the beautiful organization. Also, this theme, which was debated together, Algae as a sustainable resource for food, health, and industry, it's a very actual one. And I am convinced that all these outputs will be very important to see and to have some direction in what researches in this field will develop in the next year. Again, congratulations. Thank you very much. It was my pleasure to take part in such a high-level symposium, and good luck for all the participants. For Professor Patrick also, I wish to take this opportunity to salute and the best for all of you. Thank you.
Thank you very much. And I want to remember, this is the second edition. Next year will be the third edition.
Oh.
So we'll be here.
Yes. It's a very, very nice to have such a continuity in such an important symposium. And also, I wish you finished as soon as possible with your headquarters to be moved there in good conditions. And I'm sure that this will stimulate the our researcher, and especially young researcher, to have such a good research environment in such a new building.
Thank you. We hope to move to new headquarters soon. Mr. Patrick.
Once again, I want to wish everybody many thanks from the part of organizer of this symposium, because you spent of your very precious time, and we'll wait you for next year in the third edition of AlgaTech. Vivat, crescat, floreat, the wonder world of algae. Thank you very much.
Thank you very much.
And the beauty of the research and the beauty of such personalities present today.
Of course. Thank you very much.
Welcome.
So the last debate is the most active.
Yeah, of course.
Okay, so thank you very much. I want to remember you about the magazine. We put in the section of comment the link to our magazine, where the book will be published, the research from here. And also I want to remember to look a little bit to our YouTube page, because this event will be there with the presentation and video and everything. So thank you very much for your time, and good luck and health.
Thank you.
Bye.
Thank you.
Thank you. Bye. See you.
Thank you.
Bye.
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 Thank you.
Bye.
Bye.